All right, here we are, another Chapter 6 lecture. This lecture will be covering Concept 6.4, which deals with enzymes and their activity. Um, so let's get right in. So here we have the introduction slide. This is our Concept 6.4. Enzymes speed up metabolic reactions by lowering the energy barriers. And if you don't know what that means yet, stay tuned because you will find out in this lecture. They want you to know a little bit of vocab on this uh, slide. So the first word is a catalyst, which is a chemical agent that speeds up a reaction without being consumed by the reaction. And one form of a catalyst is an enzyme, which is a catalytic protein. And what the textbook wants you to know about an enzyme is that the magic word is a catalytic protein. So it's a macromolecule that's an enzyme. So yeah, the third bullet point tells us an example of an enzyme catalyzed reaction. And so it's the hydrolysis of sucrose by the enzyme sucrase. And if you don't remember, hydrolysis is a chemical reaction in which water is used to break down a compound. So we're breaking down sucrose into its components. And so here's figure 6.UN02, which tells us, which is a visual representation of the reaction. So here we have sucrose, and we're adding the water component, and then sucrase, the enzyme, lowers the activation energy and speeds up the reaction, which we'll get to later. And so we break it down. Hydrolysis is breaking down using water. So we get glucose and fructose, and then the water breaks apart as well and forms new bonds. So here we want to talk about the activation energy barrier, which is what I was just saying in the last slide. They want you to know in this first bullet point that every chemical reaction between molecules involves bond breaking and forming. So as we saw in the previous slide, we broke down the bonds connecting sucrose, making sucrose, which is the connection of glucose and fructose. And in addition, we broke down the bond connecting the OH and H in water. And then we formed new bonds with the OH bonding to glucose and the hydrogen molecule bonding to fructose. So formation of new bonds and breaking of the old bonds. The second bullet point wants you to know about activation energy, which is just what I was talking about. And activation energy is the initial energy needed to start a chemical reaction. And it's called the free energy of activation or activation energy. So it's the energy needed to start a reaction. And the third bullet point wants you to know that activation energy often occurs in the form of heat that reactant molecules absorb from their surroundings. So when the molecules absorb that thermal energy from their surroundings, it accelerates the reactant molecules, so they collide more often and more forcefully. It also agitates the atoms within the molecules, making the breakage of bonds a lot more likely. So that heat is really key to starting the reaction. And the textbook also comes up with this cool little metaphor about activation energy. And so they say that we can think of activation energy as the amount of energy needed to push the reactants over the top of an energy barrier or uphill, so that the downhill part of the reaction can begin. So activation energy is the energy needed to get to the pinnacle of this hill, so that the energy buildup leading up to this can start the downhill part of the reaction. And here, this is exactly what they're talking about. This is a visual representation of that metaphor. And so it's figure 6.12. It's a graph of the energy changes for a hypothetical exergonic reaction that swaps portions of two reactant molecules. And if you don't know what endergonic or exergonic reactions are, then you probably should have watched the previous Chapter 6 lectures. So this graph, they want you to know that the activation of the reactants is represented by this uphill portion of the graph. So here we have our reactants, and they're being activated on this uphill portion, probably absorbing thermal energy. And then at the summit, when this energy absorption is equivalent to the activation energy of this reaction, then the reactants begin to enter this transition state in which atoms the bonds connecting these atoms can be broken. And so then we go down the downhill portion of this reaction is represented by this energy being released. So the energy got built up, and then we were able to move into a transition state. And as they form new bonds, energy is being released, and then we get products from this energy release and from the new bondage. So what you're seeing is that the free energy starts here, increases, and then decreases to a point lower than where it was. And what this means is that the activation energy is repaid, and the formation of new bonds releases more energy than was invested in the breaking of old bonds. So you can release more energy, which allows for more chemical reactions to occur. And this slide covers how enzymes are able to speed up reactions. So instead of relying on heat, like was used in the previous example, organisms carry out catalysis in order to speed up reactions. And that's your vocab word, catalysis, which is a process by which a catalyst selectively speeds up a reaction without itself being consumed. And so the second bullet point is telling us that a catalyst, for example an enzyme, can speed up a reaction by lowering this activation energy barrier without itself being consumed. So like in that graph we saw in the previous slide, the activation area is something that needs to be reached in order for the bonds to break. And so if you lower that barrier, then less energy has to go into a reaction, which means less time is involved with that reaction. And the third bullet point is telling us that enzymes do not affect the change in free energy. Instead, they hasten reactions that would occur eventually. And here we have an animation about how enzymes work, so watch it.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
Alright, so that was a cool little video. And here is another visual representation of what they were just showing us. So this is figure 6.13, the effect of an enzyme on activation energy. And so enzymes don't affect the free energy change for a reaction. What they do is affect how much energy we need to put in, or the activation energy. So here's the reaction without an enzyme, and here's the reaction with an enzyme. So we can put less energy into it, and with the help of an enzyme, still get the same products. And we have to understand that the change in free energy, which is represented by this delta G, is unaffected by the enzyme. All right. Substrate specificity of enzymes. So this first bullet point wants us to know that enzymes are very specific for the reactions they catalyze. You can't just get any old enzyme to speed up your reaction. It requires different enzymes, and as they mentioned in the video, enzymes have three-dimensional shape, which means they choose very specific reactions to speed up. Here we have more vocab. So the reactant that an enzyme acts on is called the enzyme substrate. So this is the products. The reactants. So the reactants go into the enzyme speeds it up, and then you get the products. So the reactant that an enzyme acts on is called a substrate. And then the enzyme binds to its substrate, forming an enzyme-substrate complex. So that's just the bond between the enzyme and its substrate. And then, finally, the fourth bullet point is telling us that the active site is the region on the enzyme where the substrate binds. So that's where the bonds form between the enzyme and the substrate in the enzyme-substrate complex. And so, continuing on with this theme, enzyme specificity results from the complementary fit between the shape of the enzyme's active site and the shape of the substrate. So, enzymes require a very specific shape to bond with their specific reaction, and this is an important concept to understand. The next bullet point says that enzymes change shape due to chemical interactions with the substrate. So they are flexible, they can change shape when they bond with this substrate. And then they're telling us another vocab word, induced fit. So this induced fit of the enzyme to the substrate brings chemical groups of the active site together. So it brings the chemical groups on the bonding site together in order to help the reaction. Here we go. Here's a video to help explain all of the scary stuff I just told you about. Actually, it doesn't explain anything. It shows you the enzyme-induced fit. So here we can see, I'll play it again one more time, how the enzyme is changing shape. Moving right along. Here's another visual representation. Let me get at my highlighter. So the substrate is right here. That's our reactants that we want to speed up. And here's our large enzyme. And here's the active site. So as the substrate dilly-dallies on in to this active site, we can see that the enzyme sort of engulfs it, changes shape, and brings its chemical groups on this bondage site together. So, catalysis in the enzyme's active site. The first bullet point says that in an enzymatic reaction, the substrate binds to the active site of the enzyme, which is a concept that we just saw. The second bullet point says that the active site can lower the activation energy through four options. So it lowers the amount of energy that has to be put in order to make the reaction happen. Here's how it does it. Either by orienting substrates correctly, by straining substrate bonds, by providing a favorable microenvironment, or by covalently bonding to the substrate. And so through these four different ways, we can see how the active site is lowering the activation energy barrier in order to make the reaction happen faster. And so figure 6.15 S1 is showing us another visual representation of an enzymatic reaction. So here in part 1, substrates are entering the active site. So the substrates are the reactants. These are the people we want to break their bonds and form new bonds. So they're entering this enzyme substrate complex, and this is the part 2 is telling us that the substrates are held in active site by weak interactions. So they're bonding with the enzyme in the enzyme substrate complex. And then moving right along, we come down to part 3 where the substrates are converted to products through this reaction. So now the breaking and reforming of new bonds has occurred, and we now have the products which are still in this enzyme substrate complex here. And then, moving right along, the products are then released from the enzyme substrate complex, and then the enzyme reforms in its original shape. So notice how the enzyme was able to change shapes throughout this reaction, and then once it releases the products, it is able to then undergo this reaction again. And that's what's so important about enzymes, is that we know that they are not destroyed in chemical reactions. So, in this slide, the first bullet point is saying that the rate of enzyme catalysis can usually be sped up by increasing the substrate concentration in a solution, so the reactant concentration in that solution. And when all enzyme molecules in a solution are bonded with a substrate, the enzyme is saturated. And so at enzyme saturation, reaction speed can only be increased by adding more enzymes. So yeah, that's pretty self-explanatory. We can speed up the reaction by having more enzymes present. Here. Let's look at now the effects of local conditions on enzyme activity as we near the closing of this chapter. So an enzyme's activity can be affected by outside factors such as general environmental factors, temperature, and the pH of a solution, and also can be affected by chemicals that specifically influence an enzyme. So here we can look at effects of temperature and pH on an enzyme, and what the textbook wants you to know is that each enzyme has an optimal temperature and pH at which its reaction rate is greatest. So if you have a reaction that maybe doesn't have its best temperature when it wants a hot temperature and it's a little cooler, then the reaction won't be produced as fast as it would at that optimal temperature or pH. And so here we have a visual representation of that optimal reaction temperature or pH for certain reactions. So I could let you pause the video and examine this, because this is a really important concept to get, where optimal reactions are influenced by temperature and pH. So pause. Here are the closer up versions if you want to examine them a little more closely, but we can get to that later. And what they're talking about here with this beautiful picture of probably like sulfur springs, I don't really know, is that each enzyme has an optimal temperature and pH that favor the most active shape of the protein molecule. So that's what they want you to know right there with that picture. Moving right along to cofactors, another vocab word for you guys. Cofactors are non-protein enzyme helpers. So we know that enzymes are catalytic proteins. Cofactors are non-proteins. The second bullet point says that cofactors may be inorganic, such as a metal in ionic form, so inorganic, not carbon-based, or carbon-based organic. So an organic cofactor is called a coenzyme. So carbon-based cofactors are called coenzymes. And what they want you to know here is that most vitamins act as coenzymes, or as the raw materials from which coenzymes are made. So if you ever wondered why you need your vitamins, this is exactly why. Moving right along, enzyme inhibitors. So we have yet more vocab. So competitive inhibitors bind to the active site of an enzyme, competing with the substrate. Non-competitive inhibitors bind to another part of the enzyme, causing the enzyme to change shape and making the active site less effective. Um, Competitive inhibitors, what the textbook wants you to know, is that they reduce the productivity of enzymes by blocking substrates from entering active sites. Um, Whereas non-competitive inhibitors do not directly compete with the substrate, um, but still change shape and cause the active site to be less effective due to that shape change. So examples of these inhibitors can include toxins, poisons, pesticides, or antibiotics. And here we have figure 6.17, which is a visual representation of enzymatic inhibitors. So here we have normal binding. We see beautiful fun here, perfect space. Yeah, substrate. Sorry. And then competitive inhibition, which is where we have already something in the space that the substrate wants to bind in the active site. So the substrate's a little mad because now it's going to be hard to bond. So it makes it a little more difficult for the reaction to take place. Non-competitive inhibitors um, are on different sides, not in the active site, but change the shape, as we can see, of the active site, which causes the reaction to be a bit less effective. All right, so I believe that this is our last slide, and here's what the textbook wants you to know about the evolution of enzymes: is that most enzymes are proteins encoded by genes. Um, so your genes, DNA. Yeah. And then mutations in your genes, lead to changes in amino acid composition of this enzyme. So amino acids we know are proteins, and enzymes are proteins. So then altered amino acids in enzymes may alter the activity of enzymes, or alter the substrate specificity of an enzyme. So basically, the difference in amino acids within enzymes makes them react with different reactants, different components, and so that's what creates new enzymes to speed up new reactions, is that variation in the amino acids. And finally, the last bullet point wants you to know that under new environmental conditions, a novel form of an enzyme might be favored over the evolved form. So that's it for this lecture. Have a fun time with 6.5 and the rest of our unit. Catch you later.